as Pharmacovigilance Program of India. So that's a national program. You might have heard of malarial programs, tuberculosis program. As such, since this is very, very important topic, so it has become a national program now under the Ministry of Family Welfare. So let us see some important factors which are there in this particular topic of pharmacovigilance. Here we can see a drug that is sulfanilamide. So this is a drug which was very commonly used in 1930s, mainly in European countries, in Germany and also in US. And in US, this particular drug was commonly given for streptococcal infections. And what happened was, there were tablet forms of it and there were powder forms. Later on, there was a need of solutions, and one company, Naranjo company, produced this particular solution form. And it was distributed around 15 states, and gradually they started observing that there were many reports of deaths from this particular drug. And later on, when they found out that this particular, it is not mainly because of the active ingredient of the drug, and it was mainly because of the diethylene glycol, which is one of the constituents of this particular drug solution. So that caused around hundreds of deaths, and that came into notice after maybe around 2 to 3 months when many casualties were there. So this was mainly, it was considered as a tragedy, and we call it as sulfanilamide tragedy. So soon after this, they developed one Federal Food, Drug and Cosmetic Act, which said that there should be toxicity studies before a drug comes into the market. So that was the conclusion what they made, but still it wasn't enough. Now we could see another disaster in 1961 that was considered as thalidomide disaster. We can see here the seal limbs. So hypoplastic or aplastic limb deformities were seen in thousands of babies, and it was called as phocomelia or seedlings. And this particular incidences happened in around 1960s, when in Germany a common drug, again thalidomide, which they thought as a miracle drug and which came to the market as a sedative soon after the World War. People were suffering from depression. So insomnia was a common problem. So in those cases, this particular drug came into the market, and this was given into many of the states. Around 40 plus countries have used this particular drug, and when it came to Australia, one of the doctor, one obstetrician, he started using this, and gradually he found that it is helping in pregnant women who are suffering from morning sickness. And he started using that instead of sedative as an off-label indication. He started using in them, but the tragedy stuck. And this was not only observed in Australia, it was observed everywhere. And we can imagine the future of these babies who were born at that particular time and why it happened. So again, it is mainly the regulatory bodies which were deficient. There were deficiency in monitoring these particular aspects. So that is why there were congenital abnormalities in newborns and mothers who received thalidomide during pregnancy. So around 6,000 to 12,000 children, we can call it as, there is now a common term, there is pre-thalidomide era and a thalidomide, post-thalidomide era. So that is why post-thalidomide era is slightly vigilant over all these adverse effects. So that is why this led to introduction of stringent measures, and we have one of it as pharmacovigilance. So pharmacon means a drug, and this vigilar means it is to keep watch on. So keep watch on what? It is a drug. So this pharmacovigilance is the science and activities that is related to detection, assessment, understanding and prevention of adverse effects or any other drug related problems. So most importantly, we need to know the process, that is detection, assessment, understanding, and prevention of adverse effects. So now, why do we need it? All of us take drugs. We all of you consume many of the forms of drugs. But do we know the data behind that? So generally we think that it was given in animals, it was given in human beings, and it's coming out. So can we rely on that particular data? We know that this studies are done in very few, maybe starting with clinical phase 1 trial, which is done over 30 to 40 human volunteers. We have thousands, which is done in post-marketing surveillance. So what is the population of our world? Because drug is given in all over the world, around 7 billion people are there. Can we extrapolate the same findings? We have genetic changes, we have many racial differences are there. So we have different people. Genetic makeup is totally different when it comes. There is polymorphisms. So changes are there. Can we take the same drug which is done clinical trial in Africa, and can we compare the same thing with the Japan? It is very, 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 very difficult. So this is unreliability of this preclinical and clinical trial safety data, so which is existing, which is one of the limitations of these clinical trials, is giving us the need for this pharmacovigilance. So we have some aims and objectives which can be dealt with pharmacovigilance. That is, first one is early detection of unknown adverse drug reactions. So we need to detect. Here, two terms are important. One is early detection, other one is unknown. Known ones, it is already there in the literature. We have it in textbooks, and many of the articles give them. And when it comes to known areas, for example, if we have lignocaine, which is having 1% of anaphylaxis. So that might be in India. When it goes to Japan, there might be 10%. There might be rising frequency of these known adverse reactions also. So it is very important. Another objective of this is to detection of increased frequency of known adverse reactions also. And the next one, we need to know the possible mechanisms. So, and along with that, we need to know the benefit and the risk analysis. Only if benefit is more than the risk, we are giving that particular drug. If risk is more than the benefit, and we need to analyze this, and we need to disseminate this information all over. I have found that this particular drug has a risk. I should not keep it to myself. I have to give it to whole of the country or whole of the world. I should tell this particular thing so that signal is generated, hypothesis is generated, and they make the decision at the regulatory level either to ban the drug or continue using drug with monitoring of that particular drug. So these are some of the aims of pharmacovigilance. So early detection of unknown drugs, detection of increased frequency, and possible mechanisms we need to know, and also we need to know the risk benefit analysis and also disseminate the information all over the world. So how it can be done, we shall see. Now, we know what is pharmacovigilance. Why do we need it? We cannot rely upon these preclinical and clinical data, and we know what is required. So aims and objectives, how to go about pharmacovigilance. Then some of important terminologies which are like sound similar, like adverse effect, adverse drug reaction, and adverse event. There is difference. Adverse effect is like a general term. Any undesirable and unintended. It is not desired at all, and we don't want it. It is unintended. We do not have any intention of causing some trouble to the patient. But still, when we give the drug, any undesirable, unintended consequence of drug administration can be adverse effect. Now, we have adverse drug reaction and adverse event. Now, adverse drug reaction has the word drug also here. This is any noxious change which is suspected due to a drug. We have just the suspicion. If the drug is given at normal doses, if we are giving high doses, it becomes toxic. Since we are giving normal doses, this comes under this category adverse drug reaction, and which indicates when it is used in man, future use of the same drug, caution in the future use of the same drug, or which requires treatment or decreasing the dose of treatment. So that particular considered as adverse drug reaction. Now exclusion is expected side effects. which are already expected, are written there in the literature, it will be there on the monograph, it is there on the side effects. This is mainly for the operational definition for pharmacovigilance and some other conditions we have considered, where we are excluding side effects and poisonings. Otherwise, in adverse effects, everything is included. We have another adverse event. This is any untoward medical occurrence. We do not have a drug inside this, but it happens when we are giving a drug. So it's the medical occurrence when we are giving a drug. For example, I have given a drug in my OPD, paracetamol maybe, and the patient has met with accident somewhere near circulation. So it is not 100% that this particular accident might have happened because I have given the drug. So though the temporal relationship is there, that is time and the event, that is maybe around half an hour to 1 hour, but may not be sure. Now I have given an antihistaminic and he met with an accident. That can be because of the drug. So medical occurrence of any kind which is present when the drug or the pharmaceutical product is given but may not be related to the drug. So that can be considered as adverse event. So adverse event may not be because of the drug, adverse drug reaction definitely because of the drug, and adverse effect in general, all these terms are included under adverse effects. Now, how to tell this particular drug has caused this particular reaction? Suppose you are, I'm in my dental clinic and I have given particular drug, and someone maybe 3 or 4 days later they have developed jaundice. So am I sure that I have caused the jaundice by giving the drug, or drug has caused it, or it may be because of the disease what the person might be having? So there are chances. So we have something called as causality assessment. Now, causality assessment is the evaluation of likelihood that a medicine was causative agent of an observed adverse reaction. So we have the drug here, we have the reaction, and we need to assess the possibility that this particular reaction is because of the drug. So for that, we have some of the established algorithms given by international regulatory bodies. One is World Health Organization scale, and other one is NARANJO's algorithm. These are the common ones which are used, but there are many. CBRT scores can also be assessed. So now we know what is pharmacovigilance, we know the aims and objectives, and we have seen some of the terminologies which are important in these. Then coming to the causality assessment, this is one of the WHO, World Health Organization scale what we have. Now, as per that, when we present with adverse drug reaction form, we have certain terminologies there, whether this particular adverse drug reaction is certain or probable, possible, unlikely, conditional, or unassessable. Now, I'll go with one, not going with all of these. So for certain, if we need to call this particular reaction as a certain one, we need to have the laboratory test of normality. For example, if I given a drug and the patient has presented with jaundice, we need to have the bilirubin, direct bilirubin level mainly. So, and this should be correlated with the time that I have given the drug and it has caused some reaction. Then it should not be explained by his own disease what he is presented with. And the third one is response to withdrawal. We call it as B-challenge also. When we take out the drug, whether the reaction persists, then can we explain that pharmacologically or phenomenologically? Can we explain it is because of a specific medical disorder? So that we need to consider. The last one is the re-challenge. That is, we have stopped the drug and then we are giving it again and we are seeing whether that particular reaction occurs. So only after all these criteria are met, we call it as certain if we have the positive results. Otherwise, slightly comes down. We might call it as probable. So where there is unlikely and clinically reasonable response, so scales down until it is unassessable, where it says report suggesting of an adverse reaction cannot be judged because information is insufficient or contradictory, or data cannot be supplemented or verified. We do not have a data. So this is the WHO scale what we have, so, that is presented all over the world. Then we have another one which is called as NARANJO's algorithm. So in this, some around 9 to 10 questions are asked and the scores are given for this based on our answer is yes or answer is no and we do not know. For example, imagine you have given a drug in the OPD and the patient has come out with some reaction. First one, are there previous conclusive reports on this reaction? Have you observed before or someone else has observed? If yes, write +1, otherwise 0, or no also 0. If the adverse event occurred after the suspected drug was administered. So again, time. Again, +2 and -1. Based on this, did the adverse reaction improve when the drug was discontinued? Did the adverse reaction appear when the drug was re-administered? Based on this, we have the score, and the conclusion is made based on the score. We call it as a definite adverse drug reaction when the score is more than 9. And probable 5 to 8, possible 1 to 4, and doubtful if it is less than 4. It can be -4, varies from -4 to 30. So this is the NARANJO score we can remember, and this WHO scale that we can remember to assess the possibility that this particular drug has caused this particular reaction. Now, there are various methods of how we can monitor these particular adverse drug reactions. These are some examples: passive surveillances, stimulated reporting, active surveillances. Then we have the comparative observational studies, then targeted clinical investigation. I don't want to go in detail of this. Let me just explain at least what it is. So spontaneous reporting, I will tell you in a separate slide there, because all over the world, this is the most common form of reporting, spontaneous reporting. Then case series is just, for example, if we have around 10 reports of maybe jaundice, we have that series that we are taking and analyzing. Stimulated reporting, for example, one particular drug, maybe remdesivir, we are telling commonly pandemic it was used, and we are considering, we are motivating people by giving them the information that please watch for agranulocytosis due to remdesivir. So that becomes our stimulated reporting. Then we have the active surveillances, sentinel surveillances. In community, we are assessing how the drug is behaving, if any problems are there. They are called as sentinel sites, and that community data will be extrapolated to the whole of the country, maybe. So drug event monitoring, this is mainly prescription event monitoring. Also here, we are looking at electronic based prescriptions and looking at the reports. Then registries we have, that's also computer based. Then comparative observational studies, cross-sectional is one time, because we are just going there and looking at the point prevalences. Then case control studies, where it is a retrospective study. So we are going back, we are comparing the case and the control. Then cohort study, again, we all know we have studied these in community, dentistry also there, and it is prospective studies which are carried further and observed. Then these are some of the targeted clinical investigations also, one particular disease, one particular drug, and we are targeting and looking for the adverse drug reactions. I told you I would tell you about spontaneous reporting. Now, many of the countries have made reporting as mandatory, but in India, we haven't made it mandatory. We have many other forms of pharmacovigilance method, as I mentioned, to get the reports. One which is commonly used is the spontaneous reporting. The spontaneous reporting is the unsolicited communication by the healthcare professional to the company or the regulatory body. Unsolicited, without asking for. We are not forcing anyone. So because people have some of the troubles are there, like they are scared that if I report this particular reaction, I might be sued in court. So that would be there. Ideally, there is nothing like that. So since it is mainly because of the pharmacodynamic property of the drug, or it is mainly maybe because of unreliability of it, that is how the process is. So this is one of the charity, what we can say, almost like we are doing something good for the patient, or the country, or the world, or the mankind. So it is mainly for the patient safety. We have to do this, and that is why we have now at present, it might come in future to make mandatory for every dentist, every nurse, every healthcare professional, every doctor to report this. Soon it might be made mandatory, but for now, this is how the method is to make these spontaneous reporting. So some of the weaknesses, under-reporting is a major problem. Nobody, unless they are motivated or some acknowledgement is given, and mainly in our India, it is under-reporting itself. That is a major problem. And sensitive to selective reporting. If someone is dying because I am reporting it, I will not report thinking that it is dangerous. So I might have to give a lot of money. So that is also there. Next we have our information may be incomplete. Sometimes I might not have all the details what is required for that particular report. We have a report form, area report form, which is given by the Indian Pharmacopoeia Commission. So maybe because information is incomplete, I may not report that particular reaction. Then it depends on reporters. Yeah, definitely this is another factor. It depends upon IQ of everybody is there here. So we have all healthcare professionals, maybe different IQ levels also. I may not be able to identify that particular report, that particular reaction, maybe because of my time. So I would say, no, it is not at all. So differences are that these are some of the weaknesses what we have come across in spontaneous reporting. Then India definitely goes to spontaneous voluntary reporting. Then we have the regulatory authority in India, that is Drug Control General of India, through Indian Pharmacopoeia Commission, that is National Coordinating Center. We are dealing with this pharmacovigilance. Then in USA it is US FDA, and in Australia it is Therapeutic Goods Administration. Then the ADR forms, which is distributed in our country and all over. Many other countries they use different forms. So ideally, these are the 4 columns, very, very, very important ones, without which that ADR form is not complete. Or any adverse drug reaction processing is not complete without these. So, ADR form have suspected drug, suspected adverse drug reaction, drug we need to have, reaction we need to have, and a patient and a reporter, identifiable patient, identifiable reporter. So it cannot be a fake reporter. Otherwise, one company might be competing with another company in adverse drug reaction, and maybe my company would report the adverse drug reaction of another company. He might, that goes. So identifiable patient, identifiable reporter, suspected drug, and the event. That is very, very important in ADR forms. And these are mainly forms are distributed all over in some of the training programs, and even the sponsors are supposed to report. And this is the big boss what we have, that is World Health Organization, and UMC is the Upasala Monitoring Center. So this is the voluntary organization what we have. So mainly through World Health Organization. So it is the International Drug Monitoring. And around 1978, thalidomide tragedy is around 1961 we have observed it. After that, it has taken time. Many regulatory authorities, regulations were made, and after that, in 1978, it is established, and it's there in Uppsala, mainly Sweden. Presently, more than 131 countries nowadays, the recent data, are members of this. And in India, we have the suspected adverse drug reaction form. So mainly that is generated by CDSCO, now by Indian Pharmacopoeia Commission. So mainly by Government of India. In UK, we have the yellow card system for reporting this adverse drug reaction. And in USA, we have MedWatch form, so through which they would be reported. So this is how the flow chart I am presenting is like how it flows from a patient until again it comes back to us in a different form, or either a banned drug or as an improved version of it. So first, patient suffers with adverse effects. Suppose I'm there on the OPD, given the drug, and there is some adverse effect. So reports this particular problem. Who reports it? It's mainly a doctor, nurse, pharmacist, or can be the pharmacovigilance center. So they will collect the report, problem is discussed, and decision for the patient is made, whether to reduce the dose, stop the drug, or continue with the same with observation. That is made. So, and doctor reports problem to the local or national pharmacovigilance center, and from there it is reported through one of the software called as Vigiflow to the national center, and center reviews the problem, and any other reports of the drug will be seen. So after that, it will be sent to the VG base. So VG base is a global database what we have, and even simultaneously, center advises national regulatory authority if evidence suggests a problem. Then regulatory authority may take the decision. So, and center notifies World Health Organization through Uppsala Monitoring Committee, and Uppsala Monitoring Committee reviews the international data about the drug. They will collect all over if these particular reactions signal or hypothesis is made in some other places also, not only one particular place, and UMC notifies regulatory authorities for all members of the countries. And member countries, it will tell whether to ban the drug or continue the drug, or any changes should be made on the drug. So this is how the flowchart works, and this VG base is the global database what we have of individual case safety reports. So each report what we are collecting, it would be entered through Vigiflow, that is our domestic database what we have, and a software where we are entering these individual cases. Then from there it is going to the VG base, that is mainly monitored through World Health Organization. But and again they have the Uppsala Monitoring Committee. Around since 1968, around 16 million reports have come to the database there. And this is the Vigiflow what we have. It is a management system for recording, processing, and sharing reports of adverse effects. And it is for the domestic collection and processing of individual case study reports. And after that, provides effective means for management, review, and analysis of the national data. So this is what I was mentioning about NCC, that is National Coordinating Center. In India, we have an Indian Pharmacopoeia Commission, that is in Ghaziabad. And it's an again autonomous institution under Ministry of Health and Family Welfare to create some set standards for the drug in the country. The basic function is to update regularly the standards of drugs commonly required for treatment of disease in this region. So we have again, as I already mentioned, like malarial program or the tuberculosis program, any other national program. This program is started in 2010. That is called as Pharmacovigilance Program of India. Around 300 plus area monitoring centers are there. They collect the report and send it to the national center, from there to the WHO. So we have many peripheral, regional, and zonal centers also. The mission is to safeguard the health of the Indian population by ensuring that benefit of the use of this particular medicine outweighs the risk associated with this use. Only if benefit outweighs the risk, we are going with the drug. If risks outweigh the benefit, we are not going with these drugs. So that's very, very important to know the process, how the drug goes from the day one it is identified, maybe from a herbal product, when it comes to the molecular levels, maybe some molecular modification is made, and after that it is put on preclinical, maybe on genetics or the rats it is studied. After that it comes to the clinical phases where healthy volunteers are taken first. After that, disease well under controlled conditions is given for the patients also. Then after that it comes to the market. Again the study is done, post-marketing surveillance. Throughout now the system has come where we are detecting, assessing, and preventing these adverse drug reactions. Even after that, when it comes to the larger population all over the world, different genetic makeup, different racial conditions, different geographical areas, so different food habits. So in all other, when it is in the real world, how this particular drug behaves is, it is definitely unpredictable. For that, we need to continuously monitor this. And since it's a new branch of, it's not that new, as already seen there, it has started since 1930, this pharmacovigilance. After that, 1960, some stringent actions were taken. Later on, it has become national program in each and every country. And most commonly, we need to know what is the definition of this pharmacovigilance. And after that, we need to know what are the aims and objectives of pharmacovigilance. Then we need to know where are the centers. So national center, where is it? Then we have the international center. Then how do we assess this areas? What is the causality assessment? We need to know when do we call it as certain. So there might be a few questions on these. So causality assessment, when do we call a particular reaction as certain? Then some of the historical aspects we need to know about these two, WHO and the NARANJO's scheme interpretation. And some methods, pharmacovigilance methods. These are like research methods that we do for others. Here also for pharmacovigilance also we have the same methods. So thank you for the patience and thank you for the patient listening. So here I'm just going with these this is just a demo lecture we had to plan for before getting into per se the MCQs based questions. So, thank you all.